Satish, I was about to make fun of you, but I can't now. That was very sweet. All right. So what's happening here, right? So we have this institute. We have this $20 million. We have a directive to take these resources and the ideas, and with the support of the community here in San Francisco, I think what we need to be doing is to move these ideas very quickly into the clinic and come up with ideas that are actually going to have an impact, okay? And so that's my job. I'm going to take the basic biology that Warner's group is dealing with to come up with ideas for interventions, the knowledge gained by Mike McCune's group looking at where the virus is, and by Satish's group and how to measure it, and to actually begin to identify interventions that can be safely given to people to see if we can achieve our ultimate goal, which is to reduce the reservoir size, because we do think we need to do that. The amount of virus in the body has to be reduced. But at the same time, to manipulate the immune system so that when there's any virus left over, after we stop antiretroviral therapy, it can be controlled. And we're going to do this differently than what is traditionally done in academics, right? In academics, and you get a grant, you go into the lab, you put your head down, and you dig really, really deep. You publish a paper, and you move on. I've been in sort of a midlife crisis for the past few years. I'm sort of tired of doing that. I actually want to have an impact. And what I want to do is I want to be involved in an effort that's actually going to lead to improving health. And what we're going to try to do is we're going to use a little bit of an industry model here and merge it with the best of academics and foundations, and to try to see if we can come up with, to take the talent in this room and our collaborators outside this room to come in and to work as a team with a focused sort of Manhattan-like project to move all this type of knowledge into the clinic over the next 4 or 5 years. And I'm actually going to be spearheading a group of investigators here in San Francisco and some of our colleagues in industry, at Gilead, at the Infectious Disease Research Institute. I'll come back to them in a second, because they do know how to develop drugs, and we're going to try to actually make the impact that I just outlined. And our strategy, as Warner's sort of focusing on, has a lot to do with trying to harness the natural, innate immune responses that exist in everybody in a way that does 2 things. Number 1, forces the virus out of its hiding place, and number 2, enhances the capacity of the immune system to control that virus. Okay, and we're going to do that primarily by drugs which actually impact on dendritic cells. And this rather scary thing is a dendritic cell. It is a massive octopus that basically sits in tissues, and it's got these massive arms that reach all throughout tissue, grabs on the foreign stuff, and then when it has that foreign stuff, communicates with T cells, CD4 positive T cells and CD8 positive T cells. Okay, so that's how the immune system talks to each other. And what we're hoping to do is that these dendritic cells, which reach out and capture foreign things, will then turn on CD4 T cells, which is where the virus exists, force it out, and turn on CD8 T cells to kill that CD4 T cell. That's basically what we're doing. It's sort of a shock and kill. And the way we're going to do that is we're going to turn on dendritic cells not by exposing people to foreign things, but by exposing people to drugs that are designed specifically to turn on dendritic cells, TLR4, TLR7, these toll-like receptors that we've heard about. And that's the idea. And we think we can get away with this in large part because industry has spent the past 20 years developing these drugs, TLR agonists, to enhance vaccine responses. And some have been approved. So we have plenty of rationale that we think that we can actually take some of these stimulators, these immune system stimulators, and turn on the immune system in a safe way to do what we just said. Turn on CD4s, make the virus come out. Turn on CD8s, make the virus go away. And we think we're going to be successful. We think we have a good chance of being successful, in part based on some observations done by our group, our colleagues at Gilead, where in an animal study they showed that if you took animals, monkeys here, put them on antiretroviral drugs, So the monkeys are infected with SIV, cousin of HIV. You put them on antiretroviral drugs, their virus becomes undetectable, and then you turn on you give them a TLR agonist, these drugs that we want to develop, and boom, the virus comes out, goes back down. Then a couple weeks later, the animal comes back. Well, actually, the animal doesn't go anywhere. But the veterinarian comes back and does it again. And boom, it's like you turn it on, you turn it off. And again, and this is step one. This is getting the virus out of its hiding place. Now, the amount of virus that we're forcing out of its hiding place here, it would be, people who've been on therapy, they know what a blip is. Your virus undetectable, all of a sudden you got a blip of about 100 copies. That's what we're talking about. The virus is not spreading, it's not replicating, it's coming out. And then we think we can do this with TLR, this is TLR7 agonist. In people, we have some data, it's a bit quirky, but it's highly suggestive that we can do this in people as well with TLR9 agonists, which are actually much safer than TLR7 agonists in general. That's step one. And we think we can get away with it in large part because of these animal studies, because of the basic biology, and because we know how these drugs work in people and we know how to deliver them safely. Step two is to enhance the capacity of the immune system to control the virus. And here again, we think the TLR agonists are going to do it. These TLR agonists were designed to improve the vaccine responses. They're designed to make the immune system work better. And for me, one of the most exciting data sets out there took place when these monkeys who were on antiretroviral drugs stopped the therapy, and the monkeys who got treated with this immune stimulator, their virus did not rebound as much as the other monkeys, suggesting that we'd had to successfully turn on the capacity of the immune system to control the virus. You can do this with just one intervention, a TLR agonist that can either shock the virus out and control the virus at the same time. So these are monkey studies. They're highly provocative. And what we are going to do is we're going to work in collaboration with a foundation that is a nonprofit group that has a lot of funding, particularly from the Gates Foundation, and some of our friends from the Gates Foundation are here, but others, they've actually have a huge industry-like capacity to discover and develop for these types of studies TLR agonists, and have several that they're willing to share with us over the next few years as we try to do this in people. And right now, our plans are, over the next couple of years, to begin a series of studies in people, very careful, start with very low doses, increase the doses, looking at the combinations of various different TLR agonists and comparing that to placebo to see if we can get the virus out of its hiding place and enhance the immune system. We're not at that point where we think we can stop drugs safely, but we think we can do these types of studies in a pretty careful and very rigorous manner and ask the question pretty quickly as to whether or not this will work. And we're going to do this with these TLR agonists, TLR4, TLR7. We're going to work with a company called GeoVax and look at TLR9. But we have the capacity in San Francisco with all this expertise, and very importantly, the support of the community who want to participate in these types of studies, to answer these questions very quickly, very efficiently, and advance the data. And hopefully in the next couple of years, we'll have an answer. Either this works or it doesn't. And if it doesn't work, that's actually important as well, because then we can go on to the next backup plans that we've been working on. So that's what we'll be doing in the eliminate the eradication module that I'll be running with my colleagues. But I will just say, and I'll end in a few minutes here, but I will say that I've been doing this for 15 or 20 years. Okay, I've been doing this and a lot of people in this room know this because they participated in these studies with me, where we think we're pretty clever. We have an idea. We bring people in, we manipulate the immune system. And not once have I been successful in predicting what happens. The immune system is a big black box. And I got to tell you, at the end of the day, what we're doing is called empiric science. You do something, you measure what happens, you tell a story, and you move forward. And so we are very much open to the the idea and the concept that when you modify the immune system, that good things can happen and perhaps less good things can happen because we're turning on the immune system. We're causing inflammation, right? We do believe that this inflammatory process might be potentially harmful. In fact, and again, I won't go into any details here, but when you turn on the immune system, there's multiple pathways that happen that are hard to predict. You get T cell activation, T cell proliferation, upregulation of T regulatory cells, and so forth. And so our team is connected to about a dozen other clinical trials around the country in which the immune system is being manipulated in people, primarily to reduce inflammation. And we're going to access these samples in the amfAR Institute and use our expertise for measuring what happens to see if other pathways may seem promising. So we have a multifaceted, complementary, synergistic approach that we're going to take. First and foremost, we're going to see if we can turn on those dendritic cells to get the virus out of its hiding place with these TLR agonists, while working with our colleagues elsewhere to see if we can come up with other plans in the future in case that doesn't work. And that is the plan with our group as we move forward. And I'm going to stop there and happy to answer any questions. Thank you very much.